Okay. So I hear the word on the street is it's tough, huh?
Yeah.
Yeah. Well.
It's hard not life.
Yeah. Did you just quote Annie the musical? Ryan.
It was awesome.
Sun will come out tomorrow. All right.
We'll be back.
Muscle anatomy and phys. Muscle anatomy and phys. We have some work ahead of us today. It's the fourth time I've said it today, so I don't know. Hopefully I don't forget anything. But if you at any point today want me to go over something again, say whoa, and ask me to go over it. Or if there's something that's unclear, ask me. Don't sit there confused, because the confusion will compound and it will get even more confusing as you progress. So stop me before I get too far ahead. Okay, so today, you should be able to, by the end, explain the structure of skeletal muscle plus the function. And when you explain the function and how it works, it's going to be in a level of detail that I don't think you probably thought when you learned about it. Okay, this is one of those days that I warned you about where we're going to go to a pretty deep level, and you're going to be synthesizing material that you've learned over the course of this entire year. So if you truly have had learning take place, you'll be in great shape. You'll be able to answer all my questions. You'll be in good shape today if you can answer my questions as we go. If not, hopefully all these recording devices that I see everywhere will help you. Be prepared to make a model as well. By the end of this period, you're going to be ready to do 4.2.5, the next activity. So let's take a look at the structure of skeletal muscle. A lot of this should look like review, I would think. This is a bone. What attaches the muscle to the bone? The tendon. And then this is then the what? The skeletal muscle. But there's another term that I haven't taught you in the past. It's called, this is the body of the skeletal muscle. So the big chunk of actual muscle tissue is often referred to as the body of the skeletal muscle. So here's the body. And so now we cut it right here. None of this should be new. And it's composed of several what?
Fascicles.
The compartments are called, what again? Fascicles. So we have 3 fascicles pulled out. And now it's a good time to, I guess, talk about the coverings. What covers the muscle itself? Epimysium. Epimysium also helps form the tendon. And then what covers each fascicle?
Endomysium.
What?
Paramysium.
Paramysium. And then if I look inside of one fascicle and I pull out a muscle cell, also known as muscle fiber, it would be covered with endomysium. So reviewing the 3 tissue layers once again, what covers the outside of the whole body? Epimysium. What covers a fascicle? Paramysium. And what covers the actual muscle fiber or muscle cell itself? Endomysium. Ooh, look at this. What are these? Capillaries. Why in the heck would we need capillaries surrounding our muscle cells? Blood. Why would we need blood? Supply what? Oxygen and
Nutrients.
glucose. And what would it take away? Carbon dioxide. We'll get back to that. Okay, now here's where we go deeper. This is the muscle cell, right? Also known as what? Muscle fiber. Now we're going to go deeper. Look what I'm pulling out here. Pulling out of an individual muscle cell or muscle fiber is a myofibril. This whole thing is a myofibril. So what would this be? Myofibrils. So one muscle cell is composed of many myofibrils. Did I lose anybody yet? I haven't taught you this part, have I? All right. Now let's go deeper. I did teach you about the striations before, didn't I? What were striations? Fancy name for what? Stripes. What creates the striations? What proteins?
Actin and myosin.
Say it. Actin and myosin. Here they are right here. The myofibril is composed of 2 different types of proteins. A thin protein filament called actin. Actin is thin. Okay, actin is thin. Are we good? Myosin is thick. Okay, did I lose anybody yet? So there's only two proteins inside the muscles that we worry about. Actin and myosin. That was a lie. I'm going to contradict that in about 15 minutes. Don't hold me accountable. All right, so the actin is thin and the myosin is thick. What creates the stripes again? The striations? Actin and myosin doing what? Overlapping. So where would it be thick, the darker color? When they overlap. In the very light colors of striations? It's actually where they don't overlap and where it's only, which one do you think? Actin or myosin? Only actin. Why? Because actin is thin. So if it's a really thin protein, it'll appear lighter. So the lighter bands when we look at striations is only actin. Whereas where it's really dark, what do you think that is? It's the actin and the myosin. It's actually where they're overlapping. When they overlap, do you see how it would be darker, thicker? All right, so the darkest bands are where there's actin and myosin. All right, so now, see this blue to blue? And this blue to blue, and this blue to blue? What are these? New term. Sarcomere. S-A-R-C-O-M-E-R-E. Sarcomere. Actual definition is the contractile unit of skeletal muscle. Everybody catch that? If not, write it down. The contractile unit of skeletal muscle. What does that mean? Say it, Jaslyn. She knows the answer, but then every time I call her, no, I'm all right. The contractile unit of skeletal muscle. It's where what takes place? Contraction. So this little sarcomere, from this blue line to this blue line, is where the contraction takes place. Everything I'm going to teach you really from this point forward will have to deal with just this little area. So look what they did down below. See this line? Coming off that blue line, this line, coming off this blue line. What did they do? They zoomed in. What?
They put what's in between it.
Yeah, this is a sarcomere from here to here. Everybody see that? Notice the zigzaggy lines. Guess what we call this? Sometimes you'll see it called the Z disc. Other places you'll see it called the Z line. Do you see where they get the Z?
Yeah.
All right, it's where the actins of one sarcomere connect to the actins of another, of the next sarcomere. Because these Z lines, this would have actin going this way and actin going this way. So it's where one sarcomere connects to another. So everybody good that there would be another sarcomere over here, and there'd be another one over here? All right, and then what's in the middle? Myosin. But this myosin is thick or thin?
Thick.
It's the thick one. Look what's hanging off of here. This is new. They're called myosin heads. Myosin heads, I want you to think of like oars on a Viking boat. You ever seen like videos of Vikings rowing?
Yeah.
What do? They all do it at once, right? So imagine all those myosin heads moving at once. Guess what those myosin heads are actually going to do? They're going to pull the actins.
Closer.
What?
Closer together.
Closer. They're going to shrink the size of that sarcomere. And that's what we're going to be learning about today. Okay, so I'll pause here. Questions so far? Anyone? Did I lose anyone at all?
I need to.
Okay, good. Then let's look deeper. All right, so I have one skeletal muscle fiber here off to the side. Another name for a skeletal muscle fiber is?
Muscle cell.
Muscle cell. Good. Aren't you tired of the questions? But I figure if I ask them 10 times, maybe it'll sink in. And we're taking one myofibril out, and we're going to zoom in on it. But actually, how many myofibrils do you see here?
5.
I see 5 myofibrils. And what are the two proteins in the myofibril? Myosin and actin. We good? See this blue stuff? All right, new term. Sarcoplasmic reticulum. Reticulum. Where did we hear that before? Reticulum. Endoplasmic reticulum. Sarcoplasmic reticulum. You'll see it on the next one. All right. Oh, it's right here. It's also on the next slide that I show you. Sarcoplasmic reticulum. How would you describe its location?
On the outside.
On the outside of? A myofibril. So think of it kind of like a wrapper on a straw. If the straw is the myofibril, what is the sarcoplasmic reticulum? The wrapper. Does that help? How to envision what it's like? It covers the outside of the myofibril, but it stores something. It stores calcium ions. Do you guys know what the charge is on a calcium ion? Positive what?
1 or 2, I forget.
2. Positive 2. It's a +2 ion. So therefore, cation or anion? Cation. Somebody takes chem. So these blue things are called the sarcoplasmic reticulum, and they store what?
Calcium?
Calcium.
Ah, that's not enough people.
Calcium.
Calcium. There we go. And then this yellow is new. They're called the T tubules. The T stands for transverse. Notice, what direction would you say the T tubules run as compared to the direction of the muscle fibers themselves?
Perpendicular.
Perpendicular. So the T tubules run transverse to, or perpendicular to, the length of the muscle fiber itself. Everybody see that? 90-degree angle?
Yeah.
We'll be back to the T tubules in a little bit. Are we good on this picture? Okay, hope so. Here we go. Please realize that sarcomeres are actually round, but in this next picture, they're drawn as a two-dimensional object. Okay, so in reality, if you look at my hands, sarcomeres are more like this than like they're showing it here, which would be like this. Everybody see what I just did?
Yeah.
So I'm going to explain it in 2 dimensions, but does everybody get that it's actually 3-dimensional? I hope so. All right. So what are the black ones?
Myosin.
They're all black. That was stupid. What the myosin, the thick ones. And what are these hanging off?
The myosin head.
Myosin heads. And then I have these little things. Actin. And then what would this be called? Z-line. And then what would this be?
Another actin.
Another actin of another what? Sarcomere. So I'd have a sarcomere over here. And then what would I have over here? Another sarcomere. And so now I would like to just draw your attention to in between my hands. What's in between my hands? Only actin. At that point, that is called, has a specific name, and I'm going to give you a sarcomere diagram tomorrow. But for our purposes today, I would just like to introduce this. It's called the I band. So if you're looking at skeletal muscle under a microscope, and you see a very light-colored area, guess what it's actually called? The I band. Whereas if you look at the dark area in the center, the wide dark band, this one would be called the A band. Where there's only myosin, it's actually H band. All right, I don't expect people to have that mastered today, but I wanted to introduce it. Again, only actin is what? I band. Myosin and actin? A band. And then only myosin? H band. How do muscles work?
They shorten.
They what?
Short.
They get shorter, or they, more specifically, contract. Okay, what are the 2 protein fibers?
Myosin.
Myosin and
Actin.
actin. What if I told you neither one gets shorter?
They only slide past each other.
They slide past each other. The myosins don't change length, and the actins don't change length. So how do muscles actually work? They slide past each other. But more specifically, what do the myosin heads do?
They rotate.
They're going to take these actins and
Bring them closer.
pull them in towards the center. What would that do to the size of the H band?
Shorten it.
It would shrink it. What would that do to the size of the A band?
Shorten. Longer. Shorten.
Doesn't change it. What's the A band?
The
It stretches the length of the what?
act.
Look at it. It stretches the length of the myosin. So when you have contraction occur, the A band actually will never change size when you have contraction. I think this will become a little bit more clear in a little while. Guess what happens to your I band though?
It shortens.
It's going to shrink. Because what's happening over here in this sarcomere?
It's shrinking too.
It's shrinking. What's happening in this one over here?
Shrinking.
It's shrinking. So if they're all shrinking, what happens to the entire length of the muscle?
Contract.
It contracts. It's going to get shorter. Okay, so let's go on. Are they good? Let's see. Okay, so I have a what? Muscle fiber, also known as?
Muscle cells.
Good. And I have 1, 2, 3, 4, 5, what? Myofibrils. Each myofibril has multiple sarcomeres. See them lighten them up? So an individual muscle fiber really has like thousands of these sarcomeres end to end to end to end. Outer covering.
The myo.
In
Whoa! New term. Sarcolemma. Did you ever have this in Biology 1? Plasmalemma?
No.
All right. It's kind of a term that's fallen out of favor. They just tend to call it cell membrane. But when we're talking about specifically a muscle cell, it has a very specific name. Sarcolemma. So sarcolemma is just a very specific name for a muscle cell membrane. So what would this be made out of? What would the sarcolemma be made out of?
I'm gonna go with Evans.
What's the outer cover? What's any cell membrane made out of?
Muscle.
Phospholipid
Lipid.
bilayer. The 2 layers of fats. So this is just a fancy name for a cell membrane. Are we good? I hope so. Woo! Next. Oh, look at those! T-tubules, because they run which direction? Perpendicular. And then we see sarcoplasmic reticulum. This is not sarcolemma. It's not the cell membrane. What's inside of the sarcoplasmic reticulum? Calcium, which is what charge?
+2.
+2. Keep that in mind. We'll be back to it. In this view, though, I like to show this. What's this in here?
Same thing on that side.
It's also sarcoplasmic reticulum. So is the sarcoplasmic reticulum just on the outside?
No.
No, it surrounds the myofibril itself. Stay with me. It's an important day. And it's just going to get tougher. You have to really engage. Next one. This is kind of a pain in the butt, but I've had to, every time I get off of a website, I have to restart the Prezi. Oh well. Only have to do it 3 times. Faster, faster, faster. Now, neuromuscular junction. Look at the term. Neuromuscular junction. What is it? It's where a nerve meets a muscle. What kind of nerves are we going to be talking about then?
Neurons.
What kind of neurons? No. Sensory or motor?
Sensory. Motor.
Motor. Why? What are we making them do? What are these nerves going to do?
Move.
Make a muscle contract. So when we tell something to do something, it would be a motor neuron. So it's sending an action potential from where?
Your brain.
Brain down to a muscle. All right, so what do I have here then? What part of the nerve or neuron? Axon. What was the axon covered with? Uh-oh, crap, he's asking us stuff from 3 months ago. Yep, you should know it, you should have it down. What is the outer covering?
Myelin sheath.
Myelin sheath. What's the purpose of myelin?
To protect.
Protect and speed up because it acts as an insulator. Whoever said that? What was this area called? Here and here. Nodes of Ranvier. What is the charge of an action potential? I want an electrical value.
+30.
Positive 30. So what happened to the charge as it goes from one node to another? It weakens. Therefore, what opens up at the node? Voltage gated sodium channels. And at that point, what rushes in? And sodium rushes in until it gets to what charge? Positive 30 again. What's it called when we go from negative to positive? What was that called when we went up? Say it? Not read. Depolarization. Okay, so now going back. We have our action potential. Let's say it gets down to here. It's going to travel across the surface of the cell membrane of the neuron. And it's going to end at this end of the neuron. What do we call the endings? Axon terminals, synaptic knob, synaptic ending. I don't care what you call it. Not cleft. This is the cleft. Why? Because it refers to the space in between. The synaptic cleft is the space in between. So action potential gets down to the synaptic knob, axon terminal, whatever you want to call it. It travels across the surface of the cell membrane of this neuron. Oh, look at these. Tell me what those are. Voltage gated what? Calcium channels. So therefore, when the action potential hits it, when it hits positive 30 and it flows across this protein, these open up and what floods in?
Calcium.
Calcium ions. Whoosh, rush in. By what process? High to low. What? Diffusion. Because we have a high concentration of calcium on the outside. So calcium rushes in. Where's calcium go, Ryan? Calcium goes where? I'm just going to hang out. Hi. What?
Vesicles.
Attached to the vesicles. That's all these circles. What are the red dots? Neurotransmitters. In this case, there's a very specific neurotransmitter. You need to know it. All muscle contraction involves acetylcholine. Everybody catch that? The red dots. That's acetylcholine. A-C-E-T-Y-L. C-H-O-L-I-N-E. Acetylcholine. A-C-E-T-Y-L. Shush. C-H-O-L-I-N-E.
Can't you just call it ACH?
We can call it ACH, but I want you to know it initially because maybe in an ECA situation, they spell out acetylcholine. Okay, but the abbreviation for it properly is A, capital C, lowercase h, acetylcholine. So all these red dots are, what again? Acetylcholine. What flooded in here?
Calcium.
So calcium, when it floods in, it attaches to the vesicles, which contain what? Acetylcholine. So what do the vesicles do?
Will they merge with the
Whoa, I need a volunteer. I got too many people going. What happens to the vesicles when calcium attaches? Come on. One person. Thank you. It releases, it fuses with the cell membrane and releases its neurotransmitters into this space called what? Synaptic cleft. And then the neurotransmitters diffuse across the cleft. Where do they go?
receptor site?
These are called what?
Receptor site.
Receptor proteins. You may see them called receptor sites. What is attaching to them?
The
The acetylcholine, the ACH. But what is this down here? What is this?
Got the ending.
Where are we?
another muscle.
Neuromuscular junction. Neuromuscular junction.
Do you give up?
What is this nerve going to do? Ah! Stop. It's going to do what, Jaslyn?
Make the muscle move.
Thank you, Patricia. So you're telling me this nerve is going to make a muscle move, right? So when the nerve releases its neurotransmitter, acetylcholine, what does the acetylcholine attach to? Receptor proteins on a what? On a muscle. So, I didn't mean that. All right. So, it attaches on a muscle. These receptor proteins are on a muscle cell itself. What happens when the acetylcholine attaches? Here it's not attached. Here it's attached. Receptor proteins open. What goes in? No! Say it. Sodium. Sodium ion. Wait a minute. Sodium ions rush in. What would happen to the muscle cell at that point? What was the charge on sodium?
+2.
Positive 1. So if sodium rushes in, What's that going to do to the charge on the inside of the muscle? Make it what?
Higher.
Make it positive. What's that called? What?
Depolarization.
Depolarization. So you're telling me, let's back up the bus. Okay, quick review. Action potential. Voltage gated calcium channels open. Calcium rushes in. Calcium attaches to the vesicles, causing the vesicles to fuse with the cell membrane, releasing the neurotransmitters into the cleft. The neurotransmitter in this case? ACH, acetylcholine. It attaches to receptor proteins on the muscle cell surface. What's the cover of the muscle cell called?
Myelin.
Stop.
Something with a
Sarcolemma. It's called the sarcolemma. So you're telling me that these acetylcholine attach to receptor proteins on the sarcolemma, which is just a fancy name for muscle cell membrane. At that point, they open up, and what comes in?
Sodium.
Sodium ions. What was the charge on the sodium?
+1.
Plus 1. So that's going to make the inside of your muscles positive, which we call, what's this process called? Depolarization. So it's going to depolarize the inside of the muscle, right? Okay. Let's see if you got it.
An action potential arrives at the presynaptic terminal, causing voltage-gated calcium
You
ions.
zip your lip. All right. So the action potential comes down, opens up these voltage-gated calcium channels. What does calcium do? Attaches to vesicles. What's inside the vesicle? ACH. I pressed the wrong button.
Ion channels to open.
So after the calcium ions come in, attaches to the vesicles are filled with the ACH. Where does the ACH go?
To the cleft.
Into the cleft. It diffuses across the cleft and attaches to
The receptors.
receptor proteins. Now we're going to zoom in on acetylcholine. Acetylcholine has two components, acetic acid and choline. So let's take a look at it. We're going to attach to the receptor proteins. Boom, boom. Oh, wait, did you see what happened? Oh, that's crazy. Closed, open, closed, open, closed, open. So now wait a minute. You're telling me then when acetylcholine attaches, what happens to the protein receptors? They open, and what comes in? Sodium ions. And sodium has the positive charge, and when they come in, it makes the inside what? Positive. So the inside becomes positive, and this travels like a domino across the surface of the sarcolemma, the muscle cell membrane. And it travels all the way along the sarcolemma, and even down the T-tubule, which I'll get to in a minute. Whoa, 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 back up the bus. What's going on? We got our acetylcholine, but look, oh, it detaches. Where's the acetylcholine go? It goes over and attaches to these blue badgy looking things.
Substrate.
What are these blue badgy looking things called? What'd you say?
Substrate.
Actually, the acetylcholine is the substrate. But you're on the right track. It's an enzyme. This enzyme has a very specific name. I'll pause it right there. Everybody take a look. It's called acetylcholinesterase. Everybody see that? Acetylcholinesterase. Yeah, big fancy word. You ever think you would learn a word that long?
No.
But it ends in a what?
Ace.
ACE. Anything that ends in ASE is a what?
Enzyme.
Enzyme. Do you see how, like, it seems like we keep repeating stuff,
Yeah.
but yet it's somewhat new?
Yeah.
Today's a big synthesis day. Hopefully you're getting it. So what does acetylcholinesterase actually do? Back up the bus here. Look at it. It breaks apart acetylcholine into
To chlorine.
acetic acid and
Chlorine.
choline. Where does the acetic acid and the choline actually end up going?
Back into the muscle.
Back into,
No.
what is this? This is the axon terminal or synaptic knob, and eventually it goes back inside
Vesicles.
the vesicles. That process is called reuptake. There's a lot of drugs out there that stop that process.
What happens to your body when it stops?
When we stop that process,
Can't be reused.
acetylcholinesterase inhibitors, like there's drugs that actually inhibit that enzyme. They're used in Alzheimer's treatment. Antidepressants are selective serotonin reuptake inhibitors, so they stop the reuptake of serotonin. Some of them stop the reuptake of norepinephrine. Some of them stop the reuptake of dopamine. So we have these drugs, and they basically block that process of the neurotransmitters getting sucked back into the neuron itself. So it keeps them in the cleft longer. If they stay in the cleft longer, it's more likely that they trigger a response on the receptor protein. Did you follow that?
Yeah.
That wasn't very confident. Okay, here's the bad news.
Gotta restart the Prezi.
Yeah, but the good news, there's only one more slide.
Please press
Yeah, but this last one's going to take a while, like 12 minutes.
For one slide?
One. It's probably
Why do you say it for last?
Because it's the granddaddy.
Mine stopped recording. It was like, there's not enough room.
Okay, let's put it all together.
Okay, is it gonna be fast?
It's a picture.
No. All right, I gotta sit down for a second.
I can't press my arm.
I need you to follow the mouse because I need to restart this multiple times and explain it as I go. But I want to preface this by saying, without fail, if students come back to me after I've had them in HBS or even when I used to teach anatomy and phys, if they come back and ask for one animation, this is it. The one you're about to see. Because it explains muscle contraction better, I think, than any other one I've found. All right? If you're struggling at all, go search on Google muscle contraction animation, or YouTube even, muscle contraction animation. There's multiple good ones. But if you're going to YouTube, don't pick the one with like 314 views. Pick the one with 314,000 views. Because it's most likely a much better quality. Okay? If you don't know how to decide whether or not it's good or not, pick the one with a lot of views. All right, what are you looking at?
The
Muscle cell, muscle fiber. Therefore, it's made out of, in this case, 1, 2, 3, 4, 5, 6, 7, 8, what? Myofibrils. Myofibrils, ooh, look at this, fancy. They got what?
sarcomeres.
Sarcomeres. Here's a sarcomere, from here to here. What would I call this right here?
The
Z-line, very good. What extends from here, please watch it, to here? I-band. What extends from here to here? A-band. Good. And what's the thick filament again?
H.
Myosin.
Good, myosin. Now, what is this purple stuff? Sarcoplasmic reticulum. And then we also have T-tubules that run transverse. Okay, everybody good on the structure? All right, I have one complaint about this animation. I think the first two steps, they really take for granted that you know everything that we've talked about so far today, because they cover it in about 5 seconds. Everything that we've done today here is really covered in about 5 seconds. Ready? I'll just let you watch it.
Why don't we
It's a
just watch
quick way
this?
to go through everything.
What?
Why don't
Right.
we just watch this in the beginning?
It's not in too much detail.
You wouldn't have understood the details?
No.
Okay, what is this structure up here?
The
Synaptic terminal, and it happens to be of a mononeuron. So what are these? Vesicles. They contain what? ACH, acetylcholine. So when an action potential gets down, the acetylcholine goes into this space called the what?
synaptic cleft.
Synaptic cleft. The acetylcholine attaches to?
The receptors.
Please watch the mouse. If you don't give me the last 10 minutes, all the rest of this day was in vain. It attaches to receptor proteins. At that point, it causes those receptor proteins to open up. What comes in?
Sodium.
Sodium, causing the muscle to
Depolarize.
depolarize. Realize it happens kind of like a chain of dominoes here. Because not only does it go across the muscle cell membrane surface, the sarcolemma surface, look what else it does. It goes down a T-tubule. What's connected to the T-tubule? What's this purple? Sarcoplasmic reticulum. So now wait a minute. When I tell a muscle, contract, how does this happen? Acetylcholine. Sodium starts to rush in. Sodium rushes in here. Then do you think it comes in here?
No. Yes.
Do you think it comes in here?
Yes.
How about down here?
Yeah.
Now wait a minute. What's the charge on sodium? +1, right? But it's positive. So you're telling me that sodium will rush in by what process? Diffusion, causing depolarization, but it goes from high down to low concentration. So sodium starts to rush in. What's the charge on the sodium? +1. What was in the sarcoplasmic reticulum? What's in the sarcoplasmic reticulum? I started today by talking about it. What's stored? Calcium. What's the charge on the ion? +2. So wait a minute. Hi sodium. Hi calcium. You're +1. You're +2.
No, I don't think
What?
it's like a magnet.
Why?
Repel each other.
Positive what?
Positive and positive.
Positive and positive.
Don't mix.
They don't attach. They repel. So the positive sodium ions do what?
Repel the calcium.
Cause, what do you think these black triangles are?
Calcium.
They're the calcium ions. So the influx of the sodium cause the calcium ions to leave the sarcoplasmic reticulum. Where are they going to go?
Different.
Where's the calcium going to go? There's only one place. Huh? It was in the sarcoplasmic reticulum, which was surrounding the what? What does the sarcoplasmic reticulum cover?
Myofibril.
The what? Myofibril, which is made out of
Myosin.
myosin
Actin.
and actin. So if I kick the calcium out of the sarcoplasmic reticulum, where's it going to go?
There.
And it'll go to the what? Myosin and actin, and it'll start to surround the protein filaments. When the muscles relax, no calcium in there. No calcium at all. But when I tell a muscle to contract, what does the calcium do? Goes in, surrounds the actin and the myosin. We're ready for the next step. Let's watch it one more time before we go. See if it makes sense now. Action potential. Acetylcholine attached to receptor proteins. Depolarization. Sodium influx. Calcium goes out. Where's it go?
Into myosin.
Into surrounding the myosin and the actin. Alrighty. Let's go one level deeper.
You did not.
This is good. You ready? This is my favorite. What's the blue? Myosin, because it's thick. And then the gold spheres? Actin, because it's thick. And then I have surrounding. There's something new. And here's the people see the word and they just say, screw that, I'm not learning it. Well, don't be that person. Troponin tropomyosin complex. You see how it's wrapped around it? It's called, again, I'll repeat it. It's called the troponin tropomyosin complex. Everybody catch that? Troponin, which is this lighter green, tropomyosin, this thin protein filament, and what is it wrapped around?
Actin.
The actin. Just the actin. The troponin tropomyosin is just wrapped around the actin. What was this filament here?
Myosin.
Myosin. What's hanging off the myosin?
Myosin head.
Myosin heads. But it's not two-dimensional because, look, I got myosin heads coming this way and going this way. Because how is it? It's actually three-dimensional, more like a sphere. And so now, stay with me. Please, begging, all that is holy, stay with me. I have troponin and I have tropomyosin. Well, but my myosin heads, they're not attached yet. Why? Do you see these white spots?
Not really.
They're kind of covered up, aren't they? They're called the myosin head binding sites. So what do I need to do?
Uncover.
I need to uncover those binding sites. Guess how? Let's watch.
Some enzyme.
Let's add some calcium. Woo-hoo! Wow, did you see that?
Oh my god, do it again.
Whoa, 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 whoa. Back it up. Man, fourth time I teach it, I get slap happy, I guess. Watch where the calcium goes. Keep your eye on the troponin portion of the troponin tropomyosin complex. Calcium? Whoa! It changed the shape of the troponin tropomyosin complex. And what does it expose? The binding sites. You see how the myosin heads can grab now?
Yeah.
So if there's calcium present, it changes the shape of that troponin tropomyosin, exposing the myosin head binding sites. What can the myosin heads do? Grab. But how do the myosin heads move? Remember the Viking oars. How do they move?
In out.
Together.
What's the yellow?
The ATP.
See the ATP come in? What's the orange?
The ADP.
ADP. What's the gray?
Yeah.
A phosphorus. Where the heck do they think they're going? Where are they going? Back to the what?
Mitochondria.
Are there mitochondria in muscle cells?
Yeah. No.
A ton. There's a ton. A very high population density of mitochondria. Thank you, Jaslyn. Real high concentration of mitochondria. Why? Because muscles demand a lot of ATP. And so, why do they demand a lot of ATP? Because you need an ATP every time you want to make a myosin head go
This way.
boop. What's happening over here?
Same thing.
Same thing, but
Different.
The other way.
the other way. Same thing, but the other way. And so we're pulling them in towards the center to shorten the sarcomere as a whole. What happens when I tell my muscles to relax? I stop sending action potentials, right? So what does acetylcholinesterase do to the acetylcholine? Sucks, it recycles it, makes it go back into the synaptic knob. And so if the acetylcholine isn't there, what happens to those proteins? They close. What happens to sodium? It doesn't come in. It goes out. Okay, but... Okay, then what would happen?
Then the calcium stays.
Calcium. What calcium is in here? Holy crap! How are we going to get the calcium out? How am I going to do that? And make my muscle relax and go back to its original position. Where was the sodium? Okay, back up the bus. Calcium is stored where? Sarcoplasmic reticulum. But when sodium floods in, what does the calcium do? It rushes in and surrounds the actin and the myosin attached to the troponin, right? But then when I make my muscle relax, we got to get rid of the calcium. But the problem is, where's the sodium? It's in the what? It's in the T-tubule, but it already actually rushed into the sarcoplasmic reticulum. Because that's what, remember, the rushing in of the sodium caused the calcium to leave. How in the heck are we going to get the sodium out?
Add more calcium.
No. How are we going to get the sodium out? Huh? Think. Say it loud. Sodium potassium pump. Do you remember that from neurotransmission? So once we engage sodium potassium pump, what's the sodium do? Out. And if it leaves the sarcoplasmic reticulum, what can go back into the sarcoplasmic reticulum? The calcium ions. And then it's ready to contract again. Crazy part is, this happens in less than 1/10 of a second. Muscle contraction. It's fast.
Is that it?
Are we done?
I guess I earned my money. All right. Almost. Almost. Do you have any questions? Anything you want me to cover again? Anything you're not understanding? What? Was that as hard as you had heard?
Yeah. No, not really.
Okay, stop your recording.